All right, hello everyone. Today we will be covering chapter 20, which is the urinary system. Here are some roots, prefixes, and suffixes that you'll need to know. Most of these are foreign. We haven't really talked about any of these in past lectures. Really, the only one that we've talked about is prox, like proximal, which means next to or nearest. Renal, R-E-N, you might be familiar with as being for meaning kidney. So just become familiar with these as we go through the lecture. All right, so what makes up the urinary system? So there's really four organs. There's the kidneys. We actually have two kidneys, one on each side, and basically the function of those kidneys are to filter the blood and basically form the urine. We have the ureters. From there, the urine is going to drain down into the ureters. We have two pairs of ureters that are going to drain from each of the kidneys, and they're going to connect into the urinary bladder. And of course, we only have one bladder. And from there, that's where we store the urine. And then the urethra, at some point when it's time to go pee, the urine's going to drain from the bladder through the urethra, and you're going to go pee either in the toilet or off the balcony at 3 o'clock in the morning. All right, so here you can see that, this is the right side of the body, and this is the left side of the body. So you can see that the right kidney, we have two kidneys here. There's the right and the left, and then we have the right ureter and the left ureter, and there's the bladder here and the urethra here. You can see that the kidney, the right kidney, is situated just below the left kidney, and that's because we have the liver that kind of sits up here. So the liver kind of pushes that right kidney down just a little bit. And we have these little adrenal glands that sit on top of the kidney, and we'll talk about those later on. All right. So kidneys, they're reddish brown in color. They're kind of bean shaped. That's why we call them, kidney beans if you eat the beans. They hold about 22% of your blood supply, and they constitute less than 1% of your body weight. So they actually hold a lot of your blood compared to your total body weight. When we talk about their location, we can say that they are, you can kind of see your vertebral column here. It's kind of blocked by the vena cava and your abdominal aorta. But we can say that they lie on both sides of the vertebral column on the posterior side of the abdominal wall. And it says that they are retroperitoneal. And that just means that they lie behind that parietal peritoneum. Remember that we have the visceral peritoneum and the parietal peritoneum. The visceral peritoneum kind of holds on to the organ itself, and the parietal peritoneum lines that cavity. And we said that left kidney is slightly higher than the right kidney. Actually, we said the right kidney was lower than the left kidney, and that's due to the position of the liver. And it's surrounded by connective tissue. We call it a renal fascia, and adipose tissue, or we can also say renal fat. So here you can see the two kidneys here. Here are your kidneys, and so there's your one kidney, two kidneys. So this is your posterior or your back side. Here's your anterior or your front side. So this just kind of shows the positioning of the kidney. Here is your parietal peritoneum right here. So there's that little layer right there. So that's the retroperitoneum, or behind the peritoneum that we were talking about. All right. So more functions, with anything, it constitutes, makes the body homeostatic. Kidneys provide a lot of homeostasis or homeostatic mechanisms towards keeping the body in its normal zen or its normal functioning process. Overall, kidneys are going to, form urine and remove the urine from the body. But some of the ways that it does that is you're just going to maintain the body fluids within normal ranges. If we have, it's going to maintain that composition. So if we have, it's going to remove, metabolic wastes, excess chemicals. If we have too much whatever in excess, such as, sodium, potassium, any electrolytes, it's going to remove those. Any foreign substances, drugs, medications, any toxic metabolites. The kidney's going to filter those out and kind of remove those. And we'll talk about the difference between, there's filtration, secretion, and reabsorption, and how all that plays into the formation of urine. But it also, in maintaining body fluids within normal ranges, it's going to help adjust that blood pH. Remember that pH is basically your hydrogen ion concentration. And as your hydrogen ion concentration increases, your pH is going to decrease. As your hydrogen ion concentration decreases, your pH is going to increase. And then also blood volume. If you drink too much water, your kidneys are going to get rid of some of that water. And all of this is going to basically get dumped out in your urine. All right, some other functions of the kidneys is that it can regulate specific metabolic processes by secreting different hormones. We've already talked about erythropoietin in previous chapters. Remember, erythropoietin is that hormone that stimulates red blood cell production. We talked a couple times in 2 different chapters about how athletes abuse this. And erythropoietin is stimulated by low blood oxygen concentration. And it usually takes about 3 to 5 days for your red blood cells to for that erythropoietin to work and your red blood cells to kind of bump back up into normal ranges. So it takes about 3 to 5 days for this whole erythropoietin process to work to stimulate that red blood cell production. It also stimulates, regulates as renin. And basically renin is going to help to increase blood pressure. And we'll talk about this renin aldosterone activation system, or this renin activation system later on. Synthesis of vitamin D. Remember that active form of vitamin D and that calcitriol. We talked about that in the digestive system. Basically, that calcitriol increases calcium absorption from the small intestine. So if you're going to be on calcium supplements, chances are you're going to be on vitamin D supplements as well, because you're going to need that vitamin D to help increase or help that calcium get absorbed in from the small intestine. And then if our kidneys are not working properly, we can do these things called hemodialysis and peritoneal dialysis that kind of acts like a kidney to help filter out wastes from the blood. And we'll talk a little bit about hemodialysis and peritoneal dialysis later on. All right, so anatomically, kidney kind of looks like a bean, we've already said, but it has a convex lateral surface and a concave medial surface. Remember, concave is something that caves in on itself. So this would be, this side would be the concave medial surface, and this side is that convex lateral surface. So surrounding the kidney, it has this tough fibrous capsule called the renal capsule. And you can see this renal capsule here. There's some additional layers. We actually have this outermost layer. It's called the renal fascia. It's a tough connective tissue called the renal fascia. And then there's also this second layer called the perirenal fat capsule, which is going to help anchor that kidney in place. And then there's this third innermost layer called the renal capsule. So we're kind of starting with this third layer. So this is actually the third layer of the kidney. So that third and innermost layer is the renal capsule. And we can actually divide the kidney into three sections: the outer cortex, the middle medulla, and the inner renal pelvis. So this renal cortex is going to be the outer region. So that renal cortex is that outer region, and it's going to contain these things called renal corpuscles. And you can see these renal corpuscles here. We'll talk about these much more in depth later on. And basically, a renal corpuscle is just a bunch of capillaries and a glomerular capsule. You can see these little red things that are inside here. Those are the capillaries. And then that whole almost looks like a ball and socket joint. That socket itself is going to be that capsule itself. It's called a glomerular capsule, or sometimes you might have heard it referred to as a Bowman's capsule. But also it contains these renal columns. And it says extensions of cortex that dip into the medulla. And we'll talk about these renal columns as well. But you can see here, there's the renal medulla, there's the renal cortex, and you can see how these things kind of dip down into the renal medulla. So the renal medulla itself is the middle region, and it contains these things called renal pyramids. You can see these renal pyramids here. So these pyramid-y looking shapes. Each kidney is going to have about 8 of these pyramids. And we also call those like the kidney lobes, or the lobes of the kidneys, and they contain these renal sinus things. You can see here the renal sinuses, and a sinus is basically just kind of like a hole. So you can see like the holes that are there. And we also have fat-filled renal sinuses. And it's used for cushioning and also for urine collection. And we also have this whole structure right here is called like a hilum. And if you remember, a hilum is basically a structure, an anatomical place in an organ where a bunch of blood vessels and or nerves kind of go into that organ. So the lungs have a hilum. The spleen has a hilum. Now the kidneys have a hilum. So a hilum is an area in an organ where blood vessels, nerves, and other structures can enter and exit that organ. All right, that third structure is called a renal pelvis. It's the inner region. It's a funnel-shaped sac. So here you can see this is a very simplistic drawing. This is a renal pelvis. You can see how it's kind of like a funnel shaped here. It begins at the hilum, and it has these minor calyces and major calyces. So these minor and major calyces, basically, the urine is going to start to be formed kind of up here in these in the cortex and these medullary, these renal pyramids, and the urine is going to kind of drain down into these minor calyces. And then eventually the minor calyces the urine is going to drain down into the major calyces. And then eventually that urine is going to drain down into the ureter. And we haven't talked about the nephron yet. The nephron is actually the functional unit of a kidney. And that is where urine, that's where the whole urine production happens. So when we filter out all of the gunk out of the bloodstream, when I mentioned that we have these areas of secretion and reabsorption, that is at the nephron site. So you can kind of see here, we have that renal corpuscle, but this is actually a picture of the nephron itself. And we'll talk about the nephron in really great detail, so we're not going to cover it now. But the nephron is the functional unit of the kidney. All right, so here's like a macroscopic view or a gross view of the kidney. You can see how we have the renal artery that goes into the kidney. And then you can see the renal pelvis, the renal medulla, the renal pyramids, the renal cortex. The renal cortex, the minor calyces that are going to drain into major calyces, your renal columns, and then eventually that renal vein is going to drain out of the kidney. All right, so blood flow in and out of the kidney. So arteries carry blood into the kidney and veins carry blood out of the kidney. So we have the abdominal aorta, the heart, and we have the aorta that branches off the heart, and we have the descending aorta, and then eventually it's going to become the abdominal aorta. And that abdominal aorta, here we can see the abdominal aorta, and eventually it's going to branch off into the renal artery. And that renal artery is going to enter the kidney through that hilum, and it's actually going to transport about 15% to 30% of the cardiac output to the kidney. So remember I said that your kidney holds about 22% of your blood supply. So here, there's different values depending on what textbooks you look at. So I mentioned 22% earlier. Right now, here it says 15 to 30% of your cardiac output. So different textbooks are going to say different things. So here, CO does not mean carbon dioxide. It means cardiac output. And remember, you're going to know what cardiac output means. We discussed that in the cardiovascular system. So here is your renal artery. It comes in, and then it's going to branch off into these things called segmental arteries. And then from there, it's going to branch off even further into these interlobar arteries. And basically, these interlobar arteries just creates blood flow between the renal pyramids. And then they're going to branch off even further into these arcuate arteries, and sometimes they're called arciform arteries at the junction of the medulla and the cortex. Then we're going to branch off even further, more microscopic view, into these cortical radiate arteries. And then eventually these cortical radiate arteries are going to be going into the nephron itself. And like I said, we'll talk a lot more about the nephron. So from the nephron, so if we look, one previous slide, we have the cortical radiate arteries that are going to go into the nephron. So the cortical radiate arteries, so this would be your we'll say this is your cortical radiate artery. And then that cortical radiate artery is going to branch off into these things called the afferent arteriole that is going to enter the glomerulus of the nephron. And remember I said that you can kind of think of this as like a ball and socket joint. So here is like your socket, and that's the glomerulus or the Bowman's capsule, and I know we haven't talked about this yet. We'll talk about it later on. And this is just a bunch of capillaries that are inside there. So remember when we talked about the nervous system, we talked about afferent neurons and efferent neurons. And the definition of afferent was to go into, and the definition of efferent was to go out of. And the same concept applies to what goes into the glomerulus. Afferent arterioles are arterioles that go into the glomerulus, and efferent arterioles are what comes out of the glomerulus. So the afferent arterioles are going to be what goes into the glomerulus. And then we have this cluster of capillaries called the glomerular capillaries. We also call this the first capillary bed. And we do this because this is where filtration of blood happens. It's inside the glomerulus, inside that kind of ball and socket joint, even though it's not a ball and socket joint because, we're not talking about the musculoskeletal system or the skeletal system. There's no bones here, but I'm just using the capillaries and the nephron to kind of make you look at it as a ball and socket joint in a two-dimensional world. In reality, it's a three-dimensional world. We'll see the 3D version kind of later on. So from the glomerular capillaries, blood is going to get filtered, and that is the first step of urine formation. And we'll get much more involved in that later on. So then we have these efferent arterioles. So blood is going to leave the glomerulus through these efferent arterioles, and it's basically blood that was not filtered by the glomerulus. And then you can see how, remember, this whole structure right here is called the nephron. And then intertwining all of the nephron is this big old capillary structure. And we call those peritubular capillaries. Peri means around, and capillaries is capillaries. So this secondary capillary bed is just this capillary network that surrounds the rest of the nephron to allow for, remember I talked about secretion and reabsorption, and we'll get into the definitions of what that means later on. But we have the urine filtration in the glomerulus itself, and then in subsequent portions of the nephron, we're going to have this this secretion and reabsorption, and that's going to happen with the secondary capillary system or the peritubular capillaries. So in this section right here, this is where filtration and secretion and reabsorption is going to happen. And then from there, we're going to have the venous supply. We're going to have the venous drainage of the renal arteries, or of the kidney. And basically, they're going to parallel the arterial system. We have the efferent arterioles that are going to merge into the efferent arterioles, and then the peritubular capillaries. And then those are going to drain into the cortical radiate veins. And then they're going to drain into the arcuate veins or the archiform veins, depending on what textbook you look at. And then the interlobar veins. Now, on the arterial side, we had segmental arteries. We actually don't have any segmental veins. So from the interlobar veins, we're going to go directly into the renal veins. And then from the renal vein, the renal vein is actually going to exit the kidney through the hilum. All right, so here you can see the nephron and the blood vessels. So here you can see this right here would be that glomerulus and the first capillary bed. Here you can see the secondary capillary bed. You can see how this nephron is rather short, and this nephron is rather long. We'll talk about the difference between the two later on. But the blood supply of the kidney is rather involved. So you can see how this is just like the blood supply of the kidney. It's called a cast. So basically what they did is when they created this model, they just dumped a bunch of dye into the renal artery and then kind of let it sit for a little bit, and this is the end result. So you can see how the blood supply of the kidney is very vast. Now, here you can see how we have the all of these little bundles. These things, those are um, the glomeruli or the glomerulus, and you can see how the first capillary bed, it's very involved. They just look almost kind of looks like a brain. And then we have the afferent and efferent arterioles that come into that first capillary bed, and the efferent arterioles leave that capillary bed. And so this is just on the right-hand side. This is just a picture of the glomerulus itself. How you can see this is the glomerulus, and this is that glomerular capsule, and together they make up the renal corpuscle. So you can see the first capillary bed here, and so this is like that ball and socket joint that I was talking about, even though it's not associated with the skeletal system at all. But now we're kind of looking at it in a three-dimensional world. It's not really a ball and socket joint. It's like this encapsulated thing. And this Bowman's capsule or this glomerular capsule kind of surrounds this whole first capillary bed completely. All right, and this is just a kind of a diagram of how the blood supply flows through the kidney, and you will need to know the correct sequence of this blood supply. All right, so remember how I said that we have the short nephrons and the longer nephrons. So those are called, the short nephrons are called cortical nephrons, and the long nephrons are called juxtamedullary nephrons. So remember that the nephron is a functional unit, and we have about 1 million nephrons in each kidney. Now, the cortical nephrons, the short nephrons, make up the majority of nephrons, about 85%, and they lie almost completely within that renal cortex, just right inside that outer layer of the kidney, and they have very short nephron loops. And we haven't talked about the nephron loop, but you can see how this loop right here is actually very short. This loop is called a loop of Henle. There's different names for it, a nephron loop or a loop of Henle. I'll call it a loop of Henle, and I'll abbreviate it as LOH. All right, juxtamedullary nephrons only make up 15% of the nephrons in the kidney. They're going to have longer nephron loops. Those nephron loops actually extend down into the medulla. They extend deep into the medulla, and their renal corpuscles are going to lie kind of more deep in that renal cortex. So see how this kind of lies more superficial, and this kind of lies more deep. So the juxtamedullary nephrons, their renal corpuscle kind of lies more deep, whereas the cortical nephron, their renal corpuscle lies more superficial. And remember that a renal corpuscle is basically a glomerulus plus a Bowman's capsule. And a glomerulus is the capillary network, and the Bowman's capsule is that thing that surrounds the capillary network. And the juxtamedullary nephrons, that is going to be kind of what is important for regulating the water balance and urine concentration. All right, so the nephron itself is composed of two parts. We've already talked about the renal corpuscle, in that it is the cluster of blood capillaries and the glomerular capsule. So we have that glomerulus. The glomerulus is that capillary cluster, and it has that afferent arteriole and those glomerular capillaries and that efferent arteriole. And you can see down here that one is thicker and one is thinner. So the afferent arteriole is actually thicker and the efferent arteriole is actually thinner in diameter. So inside this glomerulus, this is where filtration happens. And like I said, we'll talk about filtration more later on. So we have blood that's going to flow into the afferent arteriole, and it's going to kind of go around this first capillary network, or this first capillary bed, or the glomerular capillary system, whatever you want to call it. And some stuff is going to get filtered out of the glomerular capillaries. And it's going to kind of get filtered out into this little space right here. And that space is called the Bowman's space. And then surrounding here is the second portion called the glomerular capsule, also known as the Bowman's capsule. And it's going to collect that filtrate from the glomerulus. So the Bowman space is that space between the capsule and the capillaries. So we have that space here, and then eventually that filtrate is going to drain kind of off that right-hand side there. And this is where urine formation begins, with filtration. Like I said, we'll talk about filtration later on. Here's another picture diagram. We have a renal corpuscle that consists of the glomerulus. And so here's your glomerulus and your capsule. That glomerular capsule or the Bowman's capsule, whatever you want to call it. We have that afferent arteriole that is thicker. So blood's going to come in that afferent arteriole, and it's going to kind of wind its way through this first capillary bed. And gunk is going to get filtered out into this Bowman space or this glomerular space. So stuff is going to get filtered out. And we've started the process of urine formation, and it's going to kind of go down this tube. And then the blood supply is going to leave through the efferent arteriole. So afferent is thicker, efferent is thinner. All right, so once the blood supply, or once for the nephron structure, we have the glomerular capsule and that Bowman space and the renal corpuscle. So that's like the first part of the nephron structure. So that renal corpuscle is that first part. The second part of the nephron is called the renal tubule. It's basically that coiled portion that we saw up here. So all of this coiled portion, the loop of Henle, the more coiled portion. So all of this coiled portion that exits that glomerular capsule, and it extends to the collecting duct. So if we go back here, this would be the collecting duct, or sometimes called the collecting tubule. Now, I will say that the nephron actually ends here. The nephron actually ends here. So this collecting tubule or collecting duct is actually not part of the nephron. It's not anatomically linked to the nephron itself, but since it still is involved in the process of urine formation. We still kind of throw it in here when we talk about the structure of a nephron, even though it's not functionally or not anatomically part of the nephron. All right, so we have this coiled tube that leaves the glomerular capsule and extends to the collecting duct. And it receives that filtrate. Remember, filtrate is what leaves the that first capillary bed and goes into that Bowman's capsule or that Bowman's space, and then it goes to a portion called the proximal tubule. Some textbooks will say proximal convoluted tubule, or PCT for short. I learned it as proximal convoluted tubule. So I will abbreviate it as PCT. Then it will go to the loop of Henle, and we have a descending loop of Henle and then an ascending loop of Henle. And then it'll go through this apparatus called the juxtaglomerular apparatus, and we'll spend some time talking about that. Then it'll go through the distal convoluted tubule, or abbreviated DCT, and then the collecting duct. And remember, I said the collecting duct is not anatomically part of the nephron, but it is functionally linked to the process of urine formation. And then the collecting duct will go through the minor calyx, then the major calyx, and it will drain into the ureters for urine formation. So here we have our Bowman's capsule. So this is our renal corpuscle. And then this is where our filtrate comes. And then here we have our proximal convoluted tubule, or our PCT. Here, our textbook just says proximal tubule, but I say proximal convoluted tubule. And then it goes into the descending loop of Henle, then our ascending loop of Henle. This is our juxtaglomerular apparatus, JGA, juxtaglomerular apparatus. That's this portion, juxtaglomerular apparatus. And then we go into the distal convoluted tubule, DCT. And then we will go into the collecting duct like we talked about earlier. So here's just another picture. So we have our Bowman's capsule with our glomerulus here, and then our proximal convoluted tubule, our descending loop of Henle, our ascending loop of Henle, and then our distal convoluted tubule here, and then our collecting duct or collecting tubule here. Remember, inside our glomerulus is our first capillary bed. Our peritubular capillaries are our second capillary bed. Now, when we talk, so this is kind of like how the structure of our nephron looks like, but when we kind of like draw the nephron as to, what gets, when we talk about, what gets reabsorbed and secreted, we actually kind of get away from this more anatomical drawing, and we kind of go more into this kind of drawing. Because it's more easy on the eye to look at in more of a stepwise process. Because when we talk about what happens in the nephron, we'll talk about the glomerulus first, then we'll talk about the proximal tubule, then we'll talk about the loop of Henle, then we'll talk about the distal tubule, and then we'll talk about the collecting duct. So it kind of is easier on the eye to look at this kind of picture as opposed to this kind of picture. All right, so now I will say I don't like this textbook and how it kind of divides up the chapter. So the juxtaglomerular apparatus. Remember that is this thing kind of right here. So now we've kind of skipped over the proximal tubule, the loop of Henle, and we're looking at this juxtaglomerular apparatus. So we kind of bypassed the ascending loop of Henle, and we have this junction of nephron back up into the glomerulus. That's the juxtaglomerular apparatus. Where that ascending limb passes between the afferent and efferent arterioles I should say in the renal capsule. Let me make sure. Actually, what I will do is in the glomerulus, I'll do this. Glomerulus of the renal corpuscle. There we go. That's what I wanted to say. All right. So at this point of contact, if we look at the cells, they're both the cells are very specialized. We have these cells called juxtaglomerular cells and the macula densa. And basically, these cells are, there's a concentration of cells. The macula densa are basically going to look at your sodium, potassium, and chloride content of the filtrate. And remember, the filtrate is what was filtered out in the glomerulus. So that first capillary system. And if you remember, sodium, salt, potassium is a salt. So anything that's salt related, think of blood pressure. So basically, the macula densa is looking at your blood pressure. And if your blood pressure is too low, it's going to secrete renin. And all renin is an enzyme that's going to help increase your blood pressure. Now, if you have too much salt, if your blood pressure is too high, it's not going to secrete renin. But your macula densa is going to look at your salt content of that filtrate, and it's going to increase or secrete renin based on your salt content of that filtrate. For your juxtaglomerular cells, it's also going to look at blood pressure as well. But the juxtaglomerular cells, what it will do is instead of secreting something, what it can do is it can adjust the diameter of your afferent arterioles. So if remember vasodilation, vasoconstriction, so if your blood pressure is too low, it can adjust the diameter of your afferent arteriole, to accommodate changes in blood pressure. And I'm not going to tell you if it's going to be vasoconstriction or vasodilation yet. We're going to talk about that in a few slides. So also this, so basically this juxtaglomerular apparatus kind of deals with blood pressure. So the macula densa is going to secrete renin, and the juxtaglomerular cells are going to look at blood pressure. So what do you think would happen if a fetus develops a macula, if there is no macula densa in its nephron. If there is no macula densa, then the fetus would not be able to detect sodium, would not be able to detect potassium, not be able to detect chloride ions, and therefore would not be able to secrete renin in response to that. So the fetus would not have renin regulation of blood pressure. So here's kind of like what I was talking about. We have the juxtaglomerular apparatus here, and that it links up with the afferent arteriole here and the efferent arteriole here. And you can see the juxtaglomerular cells that are kind of like the more ones in purple, and the macula densa are the ones that are kind of down here in the bluish color that make up the juxtaglomerular apparatus. Remember, the macula densa looks at your potassium, sodium, and chloride concentration of the filtrate. Remember, filtrate was what is filtered out. And your juxtaglomerular cells are going to adjust the diameter of the afferent arterioles in response to blood, in order to accommodate blood pressure changes or to result in blood pressure changes. It's going to vasodilate or vasoconstrict that afferent arteriole. All right, so we're going to come back to that here in several slides. All right, we're going to talk a little bit about how urine is formed first. And then we're going to kind of jump back a little bit. So urine, remember, urine is the product of nephrons and the filtrate, and it just has a lot of waste products, water, and electrolytes. And remember that we say that water and electrolytes are interdependent because the electrolytes are dissolved in water. And we have three processes of urine formation. We talked about glomerular filtration already. That is what is done by that first capillary bed inside the glomerulus itself. It's done by those glomerular capillaries. Remember that afferent arterioles come into that glomerulus, and that filtrate kind of seeps out of the those glomerular capillaries into the Bowman space. So that is filtrate. So what gets filtered out? Water and small molecules are filtered out. Large proteins, large molecules are going to remain in the blood. Just like if you remember when we talked about the that slide that I referenced several times in the lymphatic system and the cardiovascular system, we have the 20 liters that get pushed out, the 17 liters that gets pushed back in, and the 3 liters that hopefully go to the lymphatic system. The same process, the same concept applies. Large proteins don't get filtered out. Large molecules don't get filtered out. So what gets filtered out is called filtrate. And that filtrate is going to enter the renal tubules, proximal convoluted tubule, then the loop of Henle, then the descending, then the ascending loop of Henle, and then the distal convoluted tubule, and then we go through urine formation that way. So the glomerular filtration is what is filtered out. So the key concept here is first capillary bed and filtration. Filtration, I can't spell. So filtration, this is the only place where we can use the term filtration, is in the glomerulus. Now, in the different portions of the, whether it be in the different portions of the renal tubule, so in the proximal convoluted tubule, in the loop of Henle, and the distal convoluted tubule, we can have this thing called reabsorption. Now, remember, we had that peritubular capillary bed or that secondary capillary bed. All tubular reabsorption is the movement of filtered substances from the renal tubules back to the plasma. So we might filter it out in the glomerulus, but then we might throw it back into the bloodstream later on. That's all reabsorption is. We are reabsorbing whatever it is back into the bloodstream. Now, most of your reabsorption, 70% of your reabsorption, is going to be happening in the proximal convoluted tubule. And it's because the proximal convoluted tubule is lined with microvilli. Now remember, anytime we see microvilli, we think of just like the digestive system and the respiratory system, or think of digestive system. Increased surface area. Increased surface area for absorption. That is why we have microvilli there. And it's only going to reclaim the useful substances, while whatever that's going to stay in the tubule, in that proximal convoluted tubule, will continue to become urine. And we have three methods of how tubular reabsorption happens: diffusion, osmosis, and active transport. And we know diffusion. Diffusion is area of high concentration to an area of low concentration. It's a passive process. It doesn't require ATP. So we can have passive transport of lipid-soluble substances. We can have passive transport of ion channels or carriers. For osmosis, it's a passive transport of water. Sometimes it has some solutes in there. Remember the interdependence, that some ions are dissolved in water. And then active transport is going against the concentration gradient. It requires ATP. We'll talk about active transport later on. So proximal convoluted tubule, most of the time it's going to be a passive process, but 70% of reabsorption is going to happen in the proximal convoluted tubule. Now we also have tubular secretion. Tubular secretion is just the opposite of reabsorption. It's where we take things from the plasma or the peritubular capillaries and we bring them back into the renal tubules. And typically these are going to be active transport mechanisms. So it requires ATP. So some of these, or most of these, 80% is going to be the plasma that was not filtered by the glomeruli or the glomerulus. So more waste products or larger molecules. So now we're going to be adding more waste products to form the urine. So some of the substances that we'll see are going to be medications or drugs, some more ions, such as potassium and hydrogen. We already know that hydrogen is important for regulation of the pH of body fluids, and we'll talk about hydrogen ion later on. So here you can see how we have the arterial side and the venous side. We have that interstitial fluid. So the blood flow comes in, blood flow leaves. We have that peritubular capillaries. So tubular reabsorption is going from the tubular fluid to the capillaries, and tubular secretion is going from the capillaries back into the renal tubule. Now remember that This tubular reabsorption and tubular secretion can happen at any portion of the nephron, whether it be the proximal, the loop of Henle, or the distal. But remember that 70% of reabsorption is going to happen in the proximal convoluted tubule. All right, so here is kind of like the flow chart, I guess, of what you can say is going to happen. So anytime that you see filtration is that orange arrow here, that is only going to happen at the glomerulus inside that Bowman's capsule. And then we have tubular reabsorption and tubular secretion. Tubular reabsorption and secretion both happens in the proximal convoluted tubule. When we go to the descending limb of the loop of Henle, only reabsorption happens. When we get to the ascending limb of the loop of Henle, only reabsorption happens. When we get to the distal convoluted tubule, we can see that both reabsorption and secretion happens. When we get to the collecting duct, only reabsorption happens. All right, so urea and uric acid. So urea is just a byproduct of amino acid catabolism in the liver. So we have proteins. We know proteins get processed down to amino acids. When we break down proteins, we get amino acids. So this is a geeky way of saying break down of proteins. Catabolism is breaking down a byproduct of amino acid catabolism. We break down proteins into amino acids. We break down amino acids and we form urea. And in the liver, and this happens in the liver. And then the amino acids get broken down into urea in the liver, and that urea is going to enter the renal tubules. It's going to enter that proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct through filtration. So you can say that urea is filtered, but then it also undergoes both tubular reabsorption and tubular secretion. And then it says up to 80% is reabsorbed in collecting ducts and secreted in the ascending loop of Henle. So for urea, you get rid of this. So if we're talking about urea, it says it goes through glomerular filtration. So urea is going to enter here. And then it says it is reabsorbed in the collecting ducts. So it's reabsorbed in the collecting duct. So it's going to go back out into the plasma. And it says it undergoes both reabsorption and tubular secretion. So it might come back, it might go out, and it might come back in. It says up to 80% is reabsorbed in the collecting ducts and secreted in the ascending limb of the loop of Henle. So we can go, and it might be secreted in the ascending limb of the loop of Henle. And then it might be reabsorbed in the collecting duct. And you don't have to know necessarily everything of what gets filtered out, what gets reabsorbed, what gets secreted. You will need to know this one just because it's underlined. All right, so uric acid. Uric acid is a product of nucleic acid metabolism. So when we break down DNA and RNA, that's what nucleic acid is, or that's what uric acid is. It's going to enter the proximal convoluted tubule through glomerular filtration. So it's going to do the same thing. It's going to enter in through filtration. It's going to be actively transported, requires ATP. And about 10% of the uric acid is going to enter, it's going to become urine through tubular secretion, and it's excreted. Now, we have a condition called gout, and that is when we have excess uric acid in the plasma. So if we have too much uric acid that was reabsorbed, so if it was reabsorbed back into the bloodstream, we could have this condition called gout. It's excess uric acid in the plasma, and you'll see these crystal or crystalline deposits. And you'll see inflammation and pain in joints. And a lot of times people will say that you have pain in your big toe. That's a common area. And it also can be caused by eating too much seafood. Now, people who lift weights often use protein powder. So if we're going to go back up to urea now, remember, protein powder, proteins get broken down into amino acids get broken down into urea by the liver. So just the same thing is going to apply. If you're a weightlifter who uses protein powder, it's going to get filtered into the glomerular capsule and then secreted in the nephron. All right, so urine composition is going to reflect the volume of water and solutes that the kidney must excrete or conserve to maintain homeostasis. Okay, so urine composition is actually going to vary depending on your dietary intake or your physical activity. Obviously, if you drink 1 cup of water a day, you're not going to pee that much. If you drink 47 cups of water a day, you're going to pee a lot. Those are extreme examples. No one's going to drink 47 cups of water a day, hopefully not. But your urine is basically 95% water. And we've already said that your urine contains a lot of metabolic waste products. So now we can say, we talked about urea and uric acid. So we can say that urine has urea, uric acid, and also has creatinine. But we still should not see any large proteins or glucose, or you should not see any blood in your urine. With the exception, sometimes you'll see menstrual blood in your urine if you're a female, but that has nothing to do with your urinary system. That's that's the reproductive system. That comes from a different route. The blood from menstrual blood is not coming from the kidney. So if you do see blood in your urine and you're a male, or if you're not menstruating and you see blood in your urine and you're a female, you might want to go get it checked out. If you do see glucose in your urine, you're not going to see glucose in your urine. If there is a chance that you think you have glucose in your urine, you might want to go get yourself checked out by a physician or a nurse practitioner, PA, something like that. But your urine is also going to have small amounts of amino acids and some electrolytes. So your urine volume is going to be about 0.6 to 2.5 liters per day, again, depending on your dietary intake, physical activity. If you're a big runner and you sweat a lot, you might not pee as much because you're sweating and not peeing. But about 50 milliliters to 60 milliliters of urine output is normal. And we've already said that volume varies with fluid intake, sweating, body temperature, emotional state, or other environmental factors. So here you can see that there are different concentrations of electrolytes in your blood plasma, in the filtrate that comes out of your glomerulus, and what ends up in your urine. So, for example, you can see that sodium in the blood volume versus glomerular filtrate. So, pretty much all of your sodium is going to get filtered out in the glomerulus and enters that proximal convoluted tubule, but some sodium is going to be reabsorbed. Here you can see that potassium is 5 milliequivalents, 5 milliequivalents. So all of your potassium is going to get filtered out. But you can see that your urine contains a lot more potassium. So for potassium, you can say that there's a lot more potassium that's going to be secreted as that secondary peritubular capillary moves along the nephron. What's another one? So chloride, you can say that all of your chloride is going to get filtered out in the glomerulus, and then even more chloride is going to get secreted out. Phosphate is the same way. All of it's going to get filtered out, and even more is going to be secreted. Now, so here, we do not see glucose in the urine. Here you can see your urea, uric acid, and creatinine. So these are going to be your concentrations of your urine. You're not going to have to memorize these numbers. You will need to know the concentration of potassium. So you will have to know really potassium is going to be the only one. And you're not necessarily going to have to know the numbers, but you're going to have to notice that the concentration of potassium is going to be higher in the urine than it is in the glomerulus or the plasma. All right, so once the kidneys do its thing, once the nephron does its thing, we kind of have already talked about the glomerulus, the glomerular capsule, the proximal, the nephron tube, the distal tube, and now it goes into the collecting duct. So from there, the collecting duct has these openings into the renal papillae, and it's going to drain into the minor calyx. It's going to drain into the major calyx. It's going to drain into the renal pelvis, which is going to eventually form the ureter, and then the urinary bladder, and then the urethra, and then you're going to pee out the urine. All right, so back to glomerular filtration. Now, this is where we have the Bowman's capsule and the glomerular capillaries, that first capillary system. It's that first step of urine formation. We have stuff that moves from those afferent arterioles from the blood in the glomerulus into that Bowman space or that glomerular capsule. So water, small dissolved particles like creatinine, uric acid, creatine, bicarbonate, glucose, urea, amino acids, and some small ions like phosphorus, potassium, and sodium can be filtered. Now remember, large molecules such as proteins do remain in your blood because of the size. Now, if you do have large proteins in your urine, that can mean you have an improperly functioning glomerular filtration system, and you might want to go get that checked out. Now, glomerular capillaries, if you go back to blood vessels or regular blood vessels versus glomerular blood vessels or glomerular capillaries. The glomerular capillaries are more permeable than other capillaries. They actually have these things called fenestrae that are kind of tiny openings in the walls, and they allow more substances to kind of cross over or be filtered out from the capillary bed into the Bowman space. So we'll look at kind of like the fenestrae here, so we can kind of blow up this section. So you can see how there's like these holes in the, you can see these fenestrae here. You can see how there's these holes in the blood vessels that will allow substances to pass. We don't necessarily see these fenestrae in the in regular blood vessels. They're really just only in the capillary beds, and that just allows for more filtrate to be formed. Now, it says filtrate has the same composition as tissue fluid, no large protein molecules. So if you remember, when we have plasma, which is our blood, and we have that 20 liters that gets pushed out. Remember that 20 liters that gets pushed out doesn't have any large proteins. So we could say, and we call that 20 liters that gets pushed out either tissue fluid or interstitial fluid. So we can say that plasma is going to be equal to that interstitial fluid plus large protein molecules. So we can also say that this tissue fluid or this interstitial fluid is pretty much the same thing as filtrate, because filtrate is pretty much the same thing as interstitial fluid or tissue fluid, because it doesn't have those large proteins. So we could now say that plasma is equal to filtrate plus large proteins, because filtrate does not contain large proteins. All right, so filtration pressure. So we already talked about how the juxtaglomerular apparatus affects or regulates that filtration by causing vasoconstriction of the afferent, efferent arterioles. So when we look at filtration pressure, there's the equation: net filtration pressure equals forces favoring filtration minus forces opposing filtration. So forces that favor filtration would be the glomerular capillary hydrostatic pressure. We could also call that blood pressure. And this goes back to, the 20 liters and the 17 liters. Remember that that same concept applies. Forces favoring filtration is your hydrostatic pressure, and forces opposing filtration is going to be your osmotic pressure, or your protein pressure, or your colloid osmotic pressure, whatever you want to call it. So forces that oppose filtration, we could say the glomerular capillary colloid osmotic pressure. And remember, anytime we hear colloid or osmotic, we need to associate that with plasma proteins or proteins. So because we're specifically talking about glomerular capillary colloid osmotic pressure, we need to cue our brain into thinking plasma proteins in the glomerulus. And we can call it several different names, blood colloid osmotic pressure, plasma colloid osmotic, or colloid osmotic pressure. Now, I will make a statement saying that, glucose is not a protein, but because glucose does not get filtered out, glucose does contribute to osmotic pressure. So if glucose is not reabsorbed from that tubular fluid, osmotic pressure is going to increase. Remember, reabsorption is going from the paratubular, or the nephron, back into the bloodstream. So if glucose does not go back from the nephron into the bloodstream, osmotic pressure is going to increase. And also, forces that oppose filtration. We have capsular hydrostatic pressure, and we'll talk about that one in a second. So it says if the net filtration pressure in the glomerulus is positive, filtration will occur. So if this is greater than this, then we're going to filter. If this is less than this, then we're not going to filter anything out. All right, so this is kind of like the slide that we have spent several times looking at. It's not the exact same slide, but this is where our, similar to the slide, we had that 20 liters and the 17 liters, and we had that 3 liters that got picked up by the lymphatic system. That's similar to this slide. All right, so we have, now we're kind of adding a component to it. Remember, forces that favor filtration is our glomerular capillary hydrostatic pressure. So our glomerular capillary hydrostatic pressure, remember the pressure that's inside of our bloodstream is going to help push out water. So here it says it's 60, and then it says minus our plasma protein pressure, or our colloid osmotic pressure, our glomerular capillary colloid osmotic pressure, whatever we want to call it. Here it says it's 32. And then it also says that our capsular hydrostatic pressure. Now, typically, hydrostatic pressure is usually a helpful thing, and it would be under a force favoring filtration. But we already have a hydrostatic pressure that favors filtration. But now that we have liquid that is inside that capsular space, we have hydrostatic pressure that is inside the bloodstream and inside the capsular space, or inside the capsular space. So we have hydrostatic pressure in the capsular space, and we have hydrostatic pressure inside the bloodstream. So the capsular pressure the hydrostatic pressure that's inside the capsule, the Bowman's capsule or the Bowman's space, is going to push that help or possibly push that fluid, or be a force that pushes that fluid back inside the bloodstream. So that's going to be a negative pressure. So here we can see that negative pressure of negative 18. So when we add it up, we get, positive 10. And because we have here we have a positive 10, we're going to have filtration. All right, so how do we change filtration rate? So we have blood pressure changes. We can change the diameter of our afferent arterioles. We can change the diameter of our efferent arterioles. We can change this. We can change our forces favoring filtration. So we can change our glomerular capillary hydrostatic pressure. We can change our forces opposing filtration. So we can change our plasma pressure. We can change our capillary hydrostatic pressure. So we can change a lot of these things that will affect our filtration rate. So, and I know this is really confusing right now. So what I want you to do is, think of a marathon. And we have blockades on both sides of a 2-lane street the night before to prevent parking. And now there are going to be thousands of people running in that marathon, and now they have to go down this 2-lane street. And it's going to be hard for the runners to get through that blockage because of those blockades. Now, if you remove those blockades and open up the beginning of the street, more runners can pass through the street. And so now think of the runners as blood flow. If you have more runners at the beginning of the street, you're going to have more blood flow into that glomerulus. So the street is the glomerulus and the runners are going to be blood flow. If you have more blood flow into the glomerulus, you're going to have more filtration, increasing your glomerular filtration rate. So remember that we have an afferent arteriole and we have an efferent arteriole. So here is our glomerulus, and we have an afferent arteriole, we have a capillary bed, and we have an efferent arteriole. So here, if we vasodilate our afferent arteriole, if we increase that diameter, we're allowing more blood flow into the glomerulus. Now, we're not changing our efferent arteriole right now. We're just only talking about our afferent arteriole. So if all we're doing is increasing the diameter of our afferent arteriole, we're vasodilating our afferent arteriole, we're going to increase our filtration rate because we've allowed more blood to enter into the glomerulus. On the flip side, if we decrease or vasoconstrict our afferent arteriole, we're decreasing the amount of blood that can get into the afferent arteriole. Therefore, we are going to decrease the glomerular filtration rate. So this would be an example of increased blood pressure. So this is an increased blood pressure. A low blood pressure would result in a decreased glomerular filtration rate. And so you're going to need to know this table. And remember, this is just the afferent arteriole vasodilation, vasoconstriction. Basically, how much blood is in the glomerulus. All right, so now we're still going to use that marathon as an example. We have the blockade set up. The marathon runners are in that blockade and they can't go very fast. They're going slower than they want to because of that blockade. Now that 2-lane street opens up into a 4-lane road. And the marathon runners, they're not huddled together anymore, and they can run as fast as they want to. They don't have to be surrounded by people slowing them down. They can increase their speed. So again, the runners represent blood flow. So they can, and the street represents the glomerulus. So now we're talking about the efferent arterioles. So once the street opens up into that 4-lane road, the runners can kind of disperse. So now we're vasodilating our efferent arterioles. If we are vasodilating our efferent arterioles, let me draw this. So now we're not doing anything with the afferent side. We're only going to talk about the efferent side. On the efferent arterioles, if we vasodilate the efferent arterioles, we are allowing blood to leave the glomerulus faster than we normally would. So if we are allowing blood to leave the glomerulus, we are going to decrease our glomerular filtration rate. Now, if we vasoconstrict our efferent arteriole, we're going to cause that backup of blood, and we're going to force blood to stay in the glomerulus longer. Because we're forcing blood to stay in the glomerulus longer, we're going to increase our glomerular filtration rate because blood is going to stay in the glomerulus longer. So vasoconstriction of our efferent arteriole will lead to an increase in our GFR. So I hope those two concepts make sense. If not, please let me know. Here's just kind of like a picture version of what we just talked about, how we, and you can also vasodilate or vasoconstrict both the efferent and afferent arterioles. All right. So now if we put it all together, we have, our net filtration with our that's our glomerular filtration rate. That's a direct result of our glomerular filtration rate. Our forces favoring filtration is our hydrostatic pressure, and our forces opposing is our plasma protein pressure and our glomerular capsular hydrostatic pressure. And I just abbreviated all these as our, this is our glomerular hydrostatic pressure, plasma colloid oncotic pressure or osmotic pressure. And this is our glomerular capsular hydrostatic pressure, and this is our net filtration pressure, which is a direct reflection of our glomerular filtration rate. So and basically all I'm going to do is if I increase my glomerular filtration rate and I leave everything else the same, I'm going to have an increase in my glomerular filtration rate. So I increase this one and I leave everything else the same, I'm going to increase this one. In the second example, if I decrease my GHP, if I decrease that one, I'm going to decrease, and again, I'm leaving everything else the same. I'm going to decrease that. In my third example, I'm going to increase my plasma colloid osmotic pressure, and therefore I'm going to decrease my glomerular filtration rate. In the fourth example, I'm going to decrease my plasma colloid osmotic pressure. Therefore, I'm going to increase. So you can see how they kind of flip-flop from each other. So basically, if I have an increase in this, I'm going to have an increase in this. If I have an increase in this, my forces opposed, I'm going to have a decrease in my net filtration pressure. So you don't necessarily have to memorize the table, but you do have to know the concept of the formula, and you have to be able to apply the formula, which is going to give you the output of the table. So you're going to have to know what falls under forces of favoring filtration, what falls under forces opposing filtration, and know how to kind of manipulate this table. So if I said, a low blood pressure, so if I said these following statements, you would have to know that a low blood pressure would cause a decrease in your glomerular hydrostatic pressure, which would cause a decrease in your net filtration pressure, which would cause a decrease in your glomerular filtration rate. Or I could say an increase in your plasma colloid osmotic pressure or your glomerular osmotic pressure would cause a decrease in your net filtration pressure, which would cause a decrease in your glomerular filtration rate. Or I could say something like a decrease in your Glomerular capsular hydrostatic pressure would cause an increase in your net filtration pressure, which would cause an increase in your glomerular filtration rate. So that's just different ways of kind of manipulating this table. All right, so glomerular filtration rate. Basically, we already said that kidneys receive 25% of the cardiac output. We've said anywhere from 22%, 15 to 30%. Now we're saying 25%. So again, different textbooks are going to tell you different things. So it's going to be about 1.2 liters per minute. And your average adult glomerular filtration rate is about 125 milliliters per minute, or 180 liters per day. And if you take that times 2, 180 times 2 is 360. So in 2 days, your blood is going to go through your kidneys. About 360 liters worth of blood is going to go through your kidneys. So your blood plasma actually goes through your kidneys about 60 times per day. And 20% of your blood plasma is actually filtered out by the glomerulus, that first capillary bed, and goes into that Bowman's capsule. But of that filtrate, only a small portion of that is excreted as urine. So you can see that a lot of water gets filtered out at the glomerular level. So about 180 liters of water gets filtered out, but we don't pee 180 liters of water a day. We only pee about 0.6 to 2.5 liters of water. So your glomerular filtrate is 180 liters and urine is only 0.6 to 2.5 liters. So we have to have some sort of tubular reabsorption of water so that we don't get dehydrated. Because if we peed out 180 liters of water every day, we would only be able to survive about 5 minutes before we'd get killed off for dehydration, because we can't keep up with the water volume of losing 180 liters per day. Oops. All right, so renal clearance. Basically, renal clearance is the rate at which chemicals or substances are removed from the blood or plasma by the kidney. We can use this test to determine how well the kidneys are working. And we can look at it, is there glomerular damage or progression of renal disease? And the kidneys can actually take about a 90% hit before we actually start seeing signs and symptoms of renal damage. And we have this thing called, these clearance tests called inulin, not insulin, but inulin. It's basically a plant root, a polysaccharide, and it's a clearance test. We have a creatinine clearance test. But these substances, they're filtered by the glomerulus, and they are not reabsorbed and they are not secreted. So we know that if we give you a certain amount of drug or a certain amount of creatinine or a certain amount of inulin, if your kidneys are working properly, that should be in your urine, because it's not going to get it's going to get filtered, but it's not going to be reabsorbed or secreted. So we should see a 1-to-1 ratio from what we give you to what is in your urine. And you'll see, if we look at what is filtered, that filtrate passes through the afferent arterioles, the glomerular capsule, the efferent arterioles, and eventually will become urine. All right. Now, how does the glomerular filtration rate, how is it regulated? Well, we have autoregulation, and all that is a maintenance of a constant glomerular filtration rate. We can increase our systemic blood pressure by which is vasoconstriction of our afferent arterioles. We've already talked about that. We can reduce the blood flow to the glomerulus. That's going to decrease our glomerular filtration rate, and that can help maintain the kidney function. We have that juxtaglomerular apparatus, and that tubuloglomerular feedback. Remember those macula densa cells and those juxtaglomerular cells. And remember that juxtaglomerular apparatus, it can regulate that filtrate. It looks at those sodium and chloride, or the macula densa looks at those sodium chloride, and the juxtaglomerular cells looks at adjusts the diameter of the efferent arterioles to either vasoconstrict, to increase blood pressure or to decrease blood pressure. All right, so now the marathon runners that we looked at kind of back here, this is just kind of like a repeat of that table. So if we vasodilate your afferent arterioles, we're going to increase our glomerular filtration rate. What we're doing actually is we're decreasing our resistance. If we vasoconstrict our afferent arterioles, we're increasing our resistance, we're decreasing our GFR. If we vasoconstrict our efferent arterioles, remember we are causing blood to stay in the glomerulus longer. That means we're increasing our resistance because of vasoconstriction. So we're going to increase our glomerular filtration rate by keeping the blood in the glomerulus longer. So again, you're going to have to know the concepts behind this chart. So what do you think would happen to glomerular filtration rate as a result of efferent arterial vasodilation? Your GFR is going to decrease. With afferent arterial constriction, vasoconstriction, you're going to see a decrease in your GFR. What would happen to GFR as a result of a decreased blood pressure? If you have a decreased blood pressure, your GFR is going to decrease. So think of like vasoconstriction of your afferent arterioles, or think of vasodilation of your efferent arterioles. Now, what do you think would happen to your GFR as a result of increased blood pressure? Your GFR is going to increase. So think of vasodilation of your afferent arterioles or vasoconstriction of your efferent arterioles. All right, we also have hormones that are secreted by the heart. We have atrial natriuretic peptide, AMP, and BNP that can respond into increasing blood volume and pressure. Basically, they increase the excretion of sodium and therefore water. Remember, any place sodium goes, water will follow. So if I hold on to sodium, I'm going to hold on to water. If I excrete sodium, I'm going to excrete water as well. Sympathetic nervous system exerts neural control over glomerular filtration rate. Remember, your sympathetic nervous system is fight or flight. Parasympathetic nervous system is rest or digest. So if you meet the bear in the middle of the woods, you're going to want to fight the bear. Remember, we have renin, blood pressure. Your kidneys do a lot of blood pressure control. So you're going to want your sympathetic nervous system to be in control over your kidneys. So typically when we think of fight or flight, we don't care about the urinary system. We wouldn't think that the urine output is involved in fight or flight. We would expect that the parasympathetic nervous system to be in control over the urinary system, but the kidneys play a huge role in blood pressure regulation, which is a big component in fight or flight. So the sympathetic nervous system is going to win and be primary over the urinary system. So we have blood pressure changes or volume decreases. The sympathetic nervous system can cause afferent and efferent arterioles to constrict simultaneously. And we know that when afferent arterioles constrict, that's going to decrease our GFR. And when efferent arterioles constrict, that's going to increase our GFR. And overall, the net effect is going to help maintain that GFR in a normal range. All right, so more regulation of GFR. I mentioned earlier that we have that renin-angiotensin syndrome or system, or some people call it the renin-angiotensin-aldosterone system. I learned it without aldosterone, so I just call it the RAS. It's activated by low blood pressure, sympathetic stimulation, or the macula densa. Remember, the macula densa looks at the sodium, potassium, and chloride. So if we have a decrease in sodium, potassium, chloride concentration of the filtrate, so decreases in sodium concentration of filtrate causes the juxtaglomerular apparatus to secrete renin. So we have angiotensinogen. Remember, anything that's ogen is usually an inactive form. So we have to convert angiotensinogen to angiotensin 1, and renin is going to be the enzyme that does this. Then we have to convert angiotensin 1 to angiotensin 2 by this enzyme called angiotensin converting enzyme, or ACE. And so this angiotensin 2 causes vasoconstriction of the afferent and efferent arterioles, as well as the secretion of aldosterone and ADH. And over time, it will increase your thirst, retention of sodium and water, and increase your blood pressure. So if you remember from chapter 13, aldosterone retains or conserves sodium and releases or excretes potassium from the body. And ADH retains water. Remember, ADH is anti-diuretic hormone. Diuretic means to pee, so anti-pee hormone. And I forgot to say that angiotensin 2 is released by the lungs. So here we can see that angiotensinogen is released by the liver and renin is released by the kidney. So renin converts angiotensinogen into angiotensin 1. And then we have this angiotensin converting enzyme ACE that will convert angiotensin 1 into angiotensin 2. If you've ever heard of ACE inhibitors, that's the medication that we're talking about here. So ACE inhibitors would block this step, and it would help decrease your blood pressure. All right, so we've talked about aldosterone already, and how the aldosterone will retain or conserve sodium and excrete or release potassium from the body. We talked about ANP and BNP will affect the solute concentration of urine, sodium. All right, so ADH, that anti-diuretic hormone, it's going to hold on to water. So it's going to promote water reabsorption from the walls of the distal tubule and the collecting duct. So in the last part of the nephron, that distal tubule and the collecting duct, that's where ADH is going to work. Water is going to be reabsorbed by osmosis into this hypertonic medullary interstitial fluid. So we're going to have, it says hypertonic. So remember, wherever salt goes, water is going to follow. So it's going to have this medullary interstitial fluid is going to be very, very, very salt loaded. So that's why it's hypertonic. It has a high salt content. And then we're going to dump in water in hopes to neutralize that hypertonic interstitial fluid. And now, because we're taking water out of the nephron and not in the urine, that urine is now going to become concentrated and your urine volume is going to decrease. Now, if we didn't have that antidiuretic hormone, that distal tubule and collecting duct cells would not be impermeable to water, and that water would be excreted as dilute urine. And remember that we had those cortical nephrons and the juxtamedullary nephrons. They are both important in regulation of water reabsorption. However, the juxtamedullary nephrons are going to be more important in water reabsorption than the cortical nephrons are. So remember ADH, that diuretic hormone, antidiuretic hormone. So we do have a class of medications called diuretics. Lasix, Bumex, if you've ever heard that one, are those medications. They help your body eliminate more sodium. And remember, where salt goes, water will follow. And the end result is that you lose water. And that's good for people with congestive heart failure or hypertension. But caffeine and citrus juices are also classified as diuretics. So if you drink a lot of caffeine or a lot of citrus juice, they will also increase the volume of urine excreted. And then, you can also become, have very concentrated urine and low urine volume if you, let's say you take a long hot run in the summer and you don't drink enough water afterwards, or Gatorade to replenish that fluid that you've lost through sweat. You can become dehydrated that way. All right, so here we can see the effects of ADH on urine volume. So here we're going to have, let's say we have it says hypertonic interstitial or medullary interstitial fluid. So we're going to have a bunch of salt. And remember, where salt goes, water is going to follow. And to try and counteract that hypertonicity that we have. So in all this picture is saying that ADH is going to work on the distal convoluted tubule and the collecting duct. All right, here you can just kind of read this on your own. Concentration of water in the blood decreases. This is how ADH kind of works if we have blood, the fluids are too concentrated or too diluted. All right, movement of substances. So we talked about how different parts of the nephron move substances from the nephron into the capillaries, that secondary capillary system. That's reabsorption. We talked about how we can move stuff from the secondary capillary system into the nephron, or secretion. And different parts of the nephron reabsorb and secrete specific substances. And we haven't talked about what substances are reabsorbed or secreted where yet. But hydrogen ion, we know that hydrogen ion is important in regulation of pH, but hydrogen ion is actively secreted, so active transport needs ATP, is actively secreted in the entire renal tubule. So in all parts of the nephron, you're going to see active secretion of hydrogen ion. And then we've already talked about passive transport, the diffusion, passive transport of ion channels and of electrolytes through the ion channels and osmosis. All right, so movement of active transport of substances. Remember that it requires ATP and that we're going against our concentration gradient. But, we can only throw so much against that concentration gradient. So each substance has this thing called a transport maximum. We can only transport a certain number of molecules in a certain period of time, and that's called the transport maximum. And so, if you have I'm just making up numbers. Let's say you have 100 sodiums that you want to get across, but you can only do 5 sodiums every second. Your transport maximum would be 5, and you would still have an excess of sodium. And then you would have this, it says renal plasma threshold is reached when there is more transported substances in the plasma than the active transport mechanism can handle. So in the sodium case, we would have a renal plasma threshold because we can't transport as much sodium as we need to or as we would like. So we have an excess of sodium. And we actually see this when we are diabetic. If we have glucose, typically we don't see glucose in the urine. Typically we don't see, we reabsorb all of our glucose. And if we go back up to wherever that chart was, here, typically we don't see glucose in the urine. But if we are diabetic and we've had too much sugar and we can't, and we've reached our transport maximum, we have to do something with that excess glucose. And if we're diabetic, that glucose, or pre-diabetic, that glucose is going to spill into the urine. So substances that are reabsorbed by active transport, glucose is going to be in the proximal convoluted tubule, amino acids, lactic acid, citric acid, phosphorus, sodium, calcium, and sodium. All right, in the proximal convoluted tubule, remember that 70% of tubular reabsorption occurs in the proximal tubule, and that's lined with microvilli. Remember that microvilli enhances reabsorption, enhances that surface area. And about 70% of sodium is reabsorbed by active transport. And that active transport provides energy for reabsorption of other nutrients such as glucose, amino acids, different ions, vitamins, and water. Now, antibiotics are actively secreted into the proximal tubule. The key thing with antibiotics is that, and we'll talk about the aging process later on, but during the aging process, tubular functions like secretions become less efficient. So in the elderly, antibiotics will stay in the bloodstream longer than usual. So if If a normal young person takes 1,000 milligrams of an antibiotic every day, an older person might need 750 milligrams of an antibiotic. All right, so this is just you can see what happens in the proximal convoluted tubule. You don't have to know this 100%. So, we have filtration that occurs here. We've already talked about filtration, but reabsorption, remember, goes from the tubule into the bloodstream, and secretion goes from the bloodstream back into the tubules. Water is 100% reabsorbed. Sodium is 70%, glucose is 100%, 100% amino acids, bicarb is 85, uric acid is 100%, urea, phosphate, calcium, magnesium citrate, lactate, things that are secreted, bile, hydrogen, potassium chloride, drugs, toxins, uric acid, antibiotics, other medications. And again, you don't have to know the specifics. For nursing school, you might. All right, so sodium and water reabsorption. So sodium, remember we have the sodium potassium pumps that are an active process. But they also create this gradient that allows for reabsorption by secondary active transport. And if you remember from way back when we talked about the passive diffusion, osmosis, facilitated diffusion, active transport, we talked about secondary active transport. And even though it says secondary active transport, it is not active in the fact that it requires ATP. It is, even though it says secondary active transport, it is actually a passive process. It does not require ATP. So sodium is going to diffuse down the concentration gradient, and then other substances are co-transported by carrier proteins, and that glucose and negatively charged ions such as chloride or bicarb are reabsorbed by this method. So you can see that, if we look at water, we have 180 milliliters or 180 liters that are filtered, and 99% is reabsorbed, and that we pee out about 1.8 liters per day. And remember, it was like 0.6 to 2.5. So with our sodium, that's 630, 99.5% is reabsorbed. So we actually only pee out 3.2 milligrams of sodium per day. So it's important to note that when sodium ions are reabsorbed through the wall of the renal tubule by active transport, chloride ions are reabsorbed by secondary active transport, which is a passive transport, not an active transport. Secondary active transport does not require ATP, even though it says active in its name. The active part is for the sodium piece. The chloride is just along for the ride. So the chloride is, think of like a hitchhiker, which is a passive process. So if you see a test question about this, do not choose active, an active process or active transport. So with the numbers in the chart, you can see that we have about 99% of the water that is filtered out. So what do you think would happen if we did not have tubular reabsorption? I briefly mentioned this earlier when we looked at these numbers on a prior slide. We would actually have an excess loss of fluid. We would actually pee out about 180 liters per day, and we wouldn't be able to keep up with our fluid loss. All right, so here you can see how we have the sodium and water reabsorption, and that our chloride is our secondary active transport. All right, so the loop of Henle. This is kind of confusing only because it kind of works backwards. So the ascending and descending limbs, they're parallel. So where it flows, you can see the arrow that blood flows this way. And the two limbs work together. We have an ascending limb, which is on the right-hand side, or we have a descending limb that's on the left-hand side. And I put the ascending limb first, even though anatomically the descending limb is going to come first. So the ascending limb is impermeable to water. Water does not come out of the ascending limb. So you can see that only sodium and chloride do. So the ascending limb actively reabsorbs sodium and chloride, and it actively transports back into the medulla. And it results in that hypertonic interstitial medulla. Remember, that's the hypertonic that I was talking about earlier when we were talking about ADH. And that ADH comes out of the distal convoluted tubule and the collecting duct. So this ascending limb will throw out a bunch of salt. And then now the descending limb will throw out a bunch of water, hopefully to counteract that salt that the ascending limb threw out. So the descending limb is permeable to water by osmosis, but it's impermeable to salt and chloride and other ions. And in response to the ascending limb throwing out a bunch of salt, sodium, remember water is going to follow sodium, we have sodium getting dumped out of the ascending limb. So now we're going to have water getting dumped out of the descending limb to try and counteract that hypertonic fluid in the ascending limb. And then we already talked about ADH and that distal convoluted tubule and the collecting duct, and that's also going to help neutralize this osmolality or this hypertonic medullary fluid. So because of these two limbs that are kind of working together, we call it a counter-current mechanism. So this is counter-current because anatomically we go descending then ascending. But we say it's counter-current because the ascending is going to throw out salt, then the descending is going to throw out water. So this the ascending limb is going to create that hypertonic fluid by sodium moving out of the ascending limb and creating that hypertonic medullary fluid, and then that water is going to move out of the descending limb. All right, this is just another picture of that where salt is going to leave in the descending limb or in the ascending limb first, and then water is going to follow to try and counteract that hypertonic fluid. So it's the counter current because the ascending limb is going to work first before the descending limb. All right, just another picture. All right, so in the distal convoluted tubule, remember, reabsorption goes from the nephron to the bloodstream. Secretion goes from the bloodstream to the nephron. Sodium, chloride, calcium, magnesium, bicarb, and water. Secretion is potassium, hydrogen, urea, and formate. And again, you don't have to know these. All right, tubular secretion of potassium ions. We're not going to really spend any time here. All right, so this is just kind of like a chart that kind of shows what goes where. We've already talked about, the In the renal corpuscle, we have the glomerulus and the glomerular capsule. This is where we have filtrate. So we have water and dissolved substances. In the proximal tubule, we have reabsorption of glucose. That's really the only one you're going to have to know. But we've talked about all of these. We have reabsorption of proteins, reabsorption of water, reabsorption of chloride. In the distal, or in the descending loop, remember that's where we have reabsorption of water. And then the ascending limb, we have reabsorption of sodium and chloride and potassium. And then the distal tubule, collecting duct. And remember, the collecting duct is not anatomically part of the nephron, but functionally it's linked to urine formation, so we kind of keep it in there. All right, so this is just another picture of, or another slide of, again, you don't have to know this, but if you just want to know, like, let's say where bicarb is reabsorbed and secreted, or if it's secreted, bicarb, it's reabsorbed in the proximal tubule by diffusion. In the loop of Henle, it's reabsorbed by diffusion in the ascending limb. In the distal, it's reabsorbed by diffusion. And in the collecting duct, it's reabsorbed antiport with sodium. All right, so that was the kidney. That was a lot. Please let me know if you have questions. Now, moving on from the kidney itself, urine is going to flow out from the nephron into the collecting ducts. And then from here, remember, we have the minor calyces, the major calyces, the renal pelvis, the ureters, the bladder, and then the urethra. So the ureters itself, they're about 25 centimeters long, and they connect the kidney to the bladder. And they are still behind the parietal peritoneum, and they join the bladder from below. And they have 3 layers: the inner mucosal layer, the middle muscular coat, and the outer fibrous coat. The inner coat, we say, is the mucosa. The middle coat is the muscularis, and the outer coat is fibrous, or the adventitia. Remember, that inner layer is that transitional epithelium or uroepithelium. I don't know if we'll see a slide of that later on. Remember, simple squamous epithelium, pseudostratified, and there was one that was called transitional epithelium. We'll talk about that in a second. But remember peristalsis, how food moves through the digestive system through peristalsis. That's how urine gets conveyed through the ureters, through peristaltic waves. And then we know that we can have a kidney stone that can obstruct the ureter. But if we have strong enough peristaltic waves, that obstructed stone, those waves can move the stone towards the urinary bladder. And we do have this thing called the ureterorenal reflex, in which the renal arterioles constrict, and that's going to decrease urine production in the affected kidney. So if we have vasoconstriction of our renal arterioles, remember that's going to decrease the blood flow into the kidney, which is going to decrease our glomerular filtration rate. This is just a picture of the ureter itself. All right, our bladder, it's what stores the urine and sends it to the urethra. It is a hollow organ. It's distensible, meaning that it can stretch. It's a muscular organ in our pelvic cavity. It has a triangular muscle called the trigone, depending on who is pronouncing it, at the floor of the bladder. And it also contains openings at each of its 3 quarters, 2 for the ureters and 1 for the urethra. The urinary bladder also has 4 layers. The mucosa layer, which has that transitional epithelium or uroepithelium, has a submucosal layer, a muscularis layer, and a serosa layer. And it also has this thing called a detrusor muscle, which is smooth muscle fiber. And we also have an internal urethral sphincter that surrounds the neck of the bladder so that we don't pee ourselves. So here you can see the female and the male bladder. So here you can see the in the male, you can see the ureters that kind of attach to the bladder from behind. And you really, in the female, you can't see the ureter attachment, but it's going to attach from behind as well. All right. So here in the female, you can see where the ureters are going to, or I'm sorry, in the male, you can see where the ureters are going to open up into the bladder. And here's our internal sphincter. Remember, a sphincter is just like a circular, smooth muscle. All right, here's a picture of urinary bladder. Remember, this is uroepithelium or transitional epithelium. Remember, as the bladder expands, it's going to push this epithelial layer down close to its basement membrane. So I think you guys might be more familiar with this slide. So it's going to push when the bladder gets full. It's going to push the layer of cells. So here's the bladder empty, and here's the bladder full. So the bladder is empty. The cells are nice and fluffy. As the bladder fills up, the bladder is going to get all squished up, and then you pee, and then the bladder becomes empty again. The cells are all nice and fluffy, and then as the bladder fills up, they're going to get all nice and squished again. So that's uroepithelium or transitional epithelium. All right, so the urethra, it's an organ, it's a tube that connects the bladder to the outside of the body. It has kind of two layers. The inner layer is the mucus layer, the mucus coat, or the mucosal layer, and the outer layer is a thick layer called the muscularis layer. We do have two sphincter muscles. There's an internal urethral sphincter. It's involuntary. It's a bunch of smooth muscle, and we have an external urethral sphincter. That's the voluntary muscle. Remember, voluntary muscle is skeletal muscle. And then we also have a submucous coat or a submucosal that has a lot of mucus glands, like your urethral glands. Your male urethra is about 19.5 centimeters long. It has both a urination function and a reproduction function because the sperm actually migrates down the urethra as ejaculation happens. And the urethra actually has three different sections. There's the prostatic urethra, the membranous urethra, and the spongy urethra. The spongy urethra is what is found in the shaft of the penis, and it terminates in the external urethral orifice. The female urethra is much shorter than that of the males. It's only about 4 centimeters long. And that's why females are at higher risk of developing UTIs because of the short urethra, because bacteria doesn't have that far to travel to get up into the bloodstream or into the bladder. So here you can see that the female urethra is much shorter than that of the males. So for the male urethra, You can see here that there is the 3 sections. There's the prostatic urethra here. There's the membranous urethra here, and then there is the spongy urethra here. Just a cross section of the urethra. All right, so to pee, we also call it urination, or we also say micturition. And that is expulsion of urine from the bladder. And we have a reflex called the micturition reflex or the urination reflex. And that's when the detrusor muscle contracts and that internal urethral sphincter relaxes. And then also requires relaxation of the external urethral sphincter. Remember, the internal urethral sphincter is smooth muscle, so there's no voluntary control over smooth muscle. The external urethral sphincter is under voluntary control, so it's skeletal muscle. The bladder can hold as much as 600 milliliters, and we get the urge to urinate when it's about 150 milliliters. And that P reflex, or the micturition reflex, is in the spinal cord in the sacral segments. So down at the cervical, thoracic, lumbar, sacrum, the S1 to S5, those segments of the spinal cord. And we can consciously control that micturition reflex. We can say, yeah, I don't want to pee now. I don't want to pee now. Okay, yeah, I'm going to go pee now. We can consciously control that reflex by using nerve centers in the brainstem and in the cerebral cortex. So we can tell our body when it's time to pee and when it's not time to pee. Then we can also see problems with micturition. Incontinence is when we lose our ability to control micturition. If you have had a vaginal delivery, or if you lose muscle control down there, or if you have an enlarged prostate, males, if you sneeze or laugh, you might pee yourself, or have some dribble coming out. Or if you wet the bed, that's also called nocturnal enuresis. Here's just the events of micturition. Urinary bladder distends with urine. We have stretch receptors that are stimulated. Parasympathetic nerve impulses travel to the detrusor muscles. Then our brain perceives that it's time to pee. We have voluntary contraction of the urethral sphincter and inhibition of the micturition reflex. Once we decide to urinate, the detrusor muscle contracts, the internal urethral sphincter relaxes, and urine is expelled. Now, we have different abnormalities associated with the urinary system. Horseshoe kidney, nephrotic syndrome, you just kind of go on your own and figure out what these are. Renal dysplasias, abnormal kidney structure. We're not going to really talk about these. All right, kidneys, lifespan changes. As you get older, your kidneys are going to become scarred and they're grainy due to we're going to have more fibrous connective tissue that accumulates on our kidneys throughout our lifetime. We're going to start losing our kidney cells as early as 20 years old. Our kidneys are going to shrink due to loss of glomeruli, which means that our GFR is going to decrease. We've lost some of our surface area for filtration. We might develop proteins in the urine. The second part of our nephron, the tubules, might become thickened and enclosed in fat, which means reabsorption and secretion becomes less efficient. The kidneys might have a harder time clearing certain substances, and we might have bladder incontinence. All right, just some specific applications or specific diseases. Glomerular nephritis Nephritis is just inflammation of the kidney, and glomerular nephritis is inflammation of the glomerular capillaries. It can be acute or chronic. Acute glomerular nephritis, usually as a result of an abnormal immune reaction, usually caused by a streptococcus infection. Usually does not start in the kidneys, but the bacteria will migrate down into the kidneys. Basically, we're going to have these complexes, these antigen-antibody complexes are going to deposit in the glomerulus and are going to block the filtration portion of the glomerulus and not be able to clear the waste products. Then we also have chronic glomerular nephritis, and that's a progressive disease in which there's no cure. You might see more, the nephrons are going to be damaged. You're going to see hypertension, more proteins in the urine. You might see blood in the urine, more darker urine. You're going to see changes in your blood, the BUN or the blood urea nitrogen. Your creatinine levels are going to be increased. And that can lead to end-stage renal disease, in which you would need to be on either hemodialysis or peritoneal dialysis. Hemodialysis is where you have like a fistula or a graft or a catheter, and your blood is taken out of your body and filtered through a machine and put back into your body. Peritoneal dialysis is where you dump fluids into your peritoneum and it cleans your blood that way, and you dump out the fluids into a toilet or something like that. And also we can do a kidney transplant. All right, nephrotic syndrome. It's just a set of symptoms that occur in people with renal disease. Loss of plasma proteins, because now we're seeing proteins in the urine. Because we have that protein imbalance, that could lead to edema, infection, fluid accumulation. That's also edema. That protein pressure that is going to decrease. Our fluid volume is going to be adjusted. So we're going to have alterations in the renin-angiotensin system, ADH, aldosterone secretion. And what happens is that kidneys, they're going to say, oh, our blood volume is low, so let me dump out some aldosterone and ADH. So, aldosterone is conserving sodium and excreting potassium, and ADH is conserving water. And we know that with aldosterone, we conserve sodium, and wherever sodium goes, water goes. So now we're building up more fluid, which is going to make the edema even worse. All right, kidney stones. They can be composed of uric acid, nucleic acid byproducts, calcium oxalate, calcium phosphate, or magnesium phosphate. You can find kidney stones in the collecting ducts, the renal pelvis of the kidney. Kidney stones are no joke. They are very, very painful. Nausea, vomiting, see blood in your urine. A lot of times they'll pass on their own. Sometimes you need to remove them surgically. Sometimes you can do this thing called ESWL, extracorporeal shockwave lithotripsy, where you kind of sit in this bathtub and they put shock waves through this bathtub and kind of break up the stones. So get rid of your caffeine intake would be one of the big ways to prevent kidney stones. But other thing, calcium supplements, excess vitamin D, UTIs can also help, or can also create kidney stones. All right, urinalysis. Obviously, we know we go to the medical practitioner's office and we pee in a cup, and it can be used to kind of be the front line of defense to see if we have any diseases or disorders. Also check for illegal substances, but we can look at to see if there's any glucose in our urine. We have the dipstick that we can plug in or put in and kind of read the label. We'll actually be doing a urine dipstick in lab. One thing that I'll say, before we actually had the dipsticks and the lab tests to say, oh, you have diabetes, the doctor would actually stick his finger in and taste the urine, and if it tasted sweet, oh, you have diabetes. So, just be glad you're not a medical practitioner, 100 years ago, 50 years ago, whatever it was that, caused you to actually have to taste the patient's urine. And I just thought this is kind of fun. If you eat a lot of beets, it can actually turn your urine pink. And then I'm sure we all know that asparagus can cause our urine to be kind of nasty smelling. All right, so that is all I have for the urinary system. If you have any questions, please let me know. Thanks.